Yeah. Am I audible?
Yes, sir.
Okay. So there is some problem with my system for IQTL mapping, so that I will solve today. So meantime, we will complete. First, we will see how to convert the HapMap file into numerical format. Then, after that, we will see CRD and FRBD design. So, open your R Studio. One file is there called numerical HapMap. So just open that script. I will share the screen one moment. So just say this file is there, numerical HapMap. So just open that file. So before starting this all this command line, you need to set the working directory where your genotypic file is there. Wherever your genotypic file is there, so you need to set the working directory. After setting working directory, just run this library. Okay. After that, just this source command, and then this last four lines you can copy at a time and run it. So for conversion also it will give you that kinship and some other things, distribution of genotypes and everything. So this running completed. Let me know after this. So if you will check the working directory, here you will get the two files. One is Gapit genotype numerical and second one is Gapit genotype map. So now these two files are text file. So let me check whether I have shared. I will share one more command in group. How to convert this text file into Excel. That I will share in WhatsApp group. Just you can download from there. We will see that also. So in this, now already see, we have prepared a numerical files, that is numerical hap map, and this is get genotype map and genotype numerical. So how to convert it? So just open this, run the library for reader and open Excel. Then read the file. So now here, make sure the name of file should be correct. Then just save that file in Excel. So here also give same name, but the change extension is Excel. And then this last cast command. So then if you will see the directory, you will get the Excel file, Gapit genotype map. So this is the Excel file. So this is how you can convert the text files into Excel through this command line. Then secondly, now we will convert this numerical hap map. So that is also same way. Just make sure the name of the files are correct. So up to this completed. So this is the file, code file. So simply here you need to change the name of the file which you want to convert from text to Excel. That is the only change you need to make in this code file. Before that, don't forget to run the library command. So here, and one more thing, .txt is the only extension. hmp.txt is not an extension. So this complete is a file name up to this. Extensions are like .txt, .xls, .xlsx, .doc, .docx. These are the extensions. Yeah, that's what I'm saying. Before running this numerical command, here you need to set the working directory. And in that working directory, your genotypic file should be there. Then only then start the command line. Before starting these commands, you need to set the working directory. In that working directory, your genotypic file should be there compulsory.
Sir, where is
Then,
the
then
genotypic file?
ah,
Where is the genotypic file?
you have now you have two genotypic file. You can use any one.
Okay, all the files you sent, I downloaded and put in the working directory, but after running the
You can use any one file. Just copy that file and paste in one folder and set that folder as working directory.
okay.
And here, where, yeah, and in this, here just you need to change the file name. Here just you need to change the file name. Whichever file, whichever, and just you can try that. All your text files will be there. It will be converted into Excel. So now, see if I'll open this file. So this is the Excel file. So these are the accessions. These are the markers. And all your ATGC is there. We have converted into numerical data, that is 0, 1, and 2. And in second file, that map file, Here you will get the SNP, chromosome number, and the position.
Excuse me, sir.
Ah,
From where we got these files? Is this the in the file genotypic data in the today's
genotypic
folder?
data you already have, na? One small
Yes.
file and one big file. So run with small files so that you can understand easily.
Okay, sir. What you said before that one. Okay.
Yeah, old one there, that filtered file having 3000 markers.
Okay, sir.
So for QTL analysis, we need this numerical file and this map file along with phenotypic data. And by using these three files also, you can run the genome-wide association. Yesterday, day before yesterday, someone asked like numerical data is mandatory for Jivas. So it is optional, but result will be same. Just you can try with ATGC and the numbers.
Sir, what all file we need? Can you repeat, please?
These two only.
Okay.
Rest are the same files what we got during genome-wide association. This numerical data and this map data. Map means only SNP, chromosome number, and position. That's it. And by using this, you can convert your text document into Excel.
Excuse me, sir. I
Just
missed
ah,
something. So I just want to know how can we get the numerical data?
this script,
Oh, we have
numerical file having name with numerical happener.
okay. So okay. Oh, so these are the code we have to run on this data, and we will get.
Yeah, you will get the
Oh,
map file and numerical map file.
oh okay, sir.
So after running the source command,
Ah,
the next after running the source command, the next command I ran it and it gave me it gave me error.
photo.
Okay, let me send the screenshots.
Yeah, yeah. So up to this, it's fine. So this QTL analysis we will do tomorrow. First in R also, I have one script. So in R also we will do, and we will do with ICI mapping and cartographer.
So, sir, the process is if the file is unfiltered, we will filter it and then we will do what you have told today and we'll generate these two files and can
Ah,
go for GWAS or the
generally,
QTL.
Generally, HapMap file will come like 5 lakhs marker, 3 lakhs marker. So if it
Okay,
is
sir.
that much big, so you need to go for filtering.
Okay, sir.
So here, day before yesterday, we have followed the criteria for 5%. But
Sir.
sometimes what happened now, respective supervisors or respective in charges will tell like go for 1% also. So that 1 or 5% it will differ.
Okay, sir.
Either 1% or 5%. Either or it will be differ.
Okay, sir.
Okay. So now after this, we will take some different road and we'll change the topic like field data.
Sir, which one is the genotype file?
You have two files. One is with 3042 markers and other is with named GBS tassel. So any file you can use,
Okay.
but make sure the names of the file, your actual file name and the file names in script. So everything should be whichever file you are working, so that file should be there in your working directory first. Without that, your commands will not run. So he will delete this. This is becoming messy. So yeah. So now no need of this. Yeah. So this is your FRBD design data. So what it mean? What is FRBD?
Factorial.
Factorial RBD.
Okay. In which cases we will use it?
In land condition, the soil fertility gradient from gradually it start from one direction to another direction, sir. Heterozygous field condition.
Okay.
When you
So
have
here,
more than when you have more than two factors
okay. So
and
here, how many
different
factors?
levels,
Here, how many factors you are observing?
we have three factors.
Okay. Which are
Yes,
those?
sir.
We have genotype as factor one with different
Okay.
levels. Then we have treatment with different levels. Then we have salinity in
Okay.
different levels.
Yeah. So we have these three factors, three treatments, okay, and three replications. So we have two different levels of salinity, three different treatments. The applications are three. And characters are 11.
So replication is also considered as a factor only, right?
Yeah, it will replication also it will consider as one of the factor, but our main factors are three. That is, we need to see the interaction between genotype treatment, treatment salinity, and genotype and salinity. So if you will see the command lines, replication is also considered as one of the factors, but the major factors are these three.
And excuse me, so what is the difference in the different treatment?
So here 1 minute, I will tell you the details. So these treatments are based on the biochemical properties. Actually, this data belongs to okra crop. Seed priming of okra. So during that seed priming experiment, that particular person induces the salinity at two different levels. So genotype, three different treatments, and salinity is one of the factors. And these are the 11 characters. What is criteria means what, Raj?
So criteria means like for salinity there are two parts as S0 and S1. For replication
Ah,
also there is R1, R2, and R3. So how we are
ah.
considering that replication should not be considered as a factor, and we have to consider only genotype treatment and salinity as a factor only. But in RBD we were selecting replication also as a factor. There also replication was three replications was their maximum.
Then RBD, if you will not consider replication and as a factor, then how will you interpret the significant, non-significant genotypes? Suppose in RBD, I am not interested in replication. Then tell me how will you conclude?
So in factorial RBD, sir, replication doesn't play any role, sir.
What I told,
Okay, sir.
Sir, for that,
replication is there. Ah,
sir, for that one formula is there, sir. For RBD, T minus 1 into R minus 1 should be equal to or more than 12, sir.
yeah,
So we have
yeah.
to count for CRD means T into R minus 1, but for RBD, T minus 1 into
T
R minus 1
minus 1 into R minus 1.
R minus 1. Yes, sir.
So
Treatment
effect
degrees of
is,
freedom into replication degrees of
yeah,
freedom.
so that's why replication effect will be minimum in FRBD.
Okay, sir.
So if you will open this script, so just I think here no need to edit anything with this data except just check the file names. And if you are importing the data from import tab, please avoid this command, highlighted command. Please avoid that if you are importing the data through R. If you want to import the data through command line, then don't forget to set the working directory. And don't forget to change here the name of the file. So see, I will also, this time I will import the data through command line. Okay, then so here in this command, we are giving the observations, trait column. And now, by one, here we are giving the name of the output file. Then, after that, the plots, interaction plots will be there. So, likewise, whenever you will use the different file name and different traits, so you need to edit this. Then we need to save whatever we have analyzed. So after completing this, if you will check your working directory, you will get the diagrams in PDF. So here you can see interaction of genotypes and treatments for this particular trait. Then genotype salinity and treatment salinity. These three graphs for each and individual trait. And then you will get one text file with FRBD analysis of variance. Then after this,
Hello, sir.
Yes, tell me.
Yeah, so I was looking at the ANOVA table. So I saw that the three-way interactions are significant, the genotype, treatments, and salinity. So for trait C1, so how can we visualize this? Maybe to plot it graphically to show the interaction between genotype, treatment, and salinity.
So graphical interactions, here it is there.
No, this is between genotype and treatment. I'm talking about the interaction between the three factors.
So it is actually between the two factors, any two, that is genotype and treatments first, then genotype and salinity. So you can interpret this from all these three.
Sir, after running library, we have to set the directory and then we have to paste that script, no, sir?
Which statistic?
No, sir, script, sir, that script which you told that if we don't import it from Excel, we can just paste it step one, sir. So before that only we have to set the working directory, sir.
Working directory you need to set every time. Before
Okay, sir.
running only you will set, na? After running, how what is the use? So after that, we will plot the mean values for all the traits. Here you need to go individually, trait wise. Here I have selected C10.
Hello, sir.
Yes.
Sir, when I'm running that data, means first parameter, first code only, it is showing me error in use method mutate. I have shared the screenshot to you as well.
1 minute. Which code?
This after step one, first data I haven't run that because I have imported from the R only. Then second
Yeah.
data.
So have you observed any change here? Is there anywhere written your file name is there in this highlighted command?
No, sir.
Then instead of your file name, which name is there? In place of your file name, which name is there?
Is it muted or data? Data after data one.
Yeah. So if you are, that's why I didn't tell this thing. I'm expecting from you only, you people only. So if you are importing through R, you need to annotate your FRBD file with the name data. So see here this line.
Okay,
So
sir.
this highlighted command is putting that.
Yes.
Highlighted command is quoting that we need to import this data underscore experiment to file with name data.
Okay, sir. Understood.
Yeah. So like this, graphs will come from the FRBD mean value over the two different levels of salinity. Across the three treatments for S0, three treatments for S1. And these all are the genotypes.
And this analysis for the C10, the trait number 10. Correct?
Yeah, so individually you need to draw the graph for all the 11 characters.
Okay, and what the command we have right now, that is common for all, and so we have to just put like 1, 2.
Yeah, so this is the command for graph. So here you need to change the character name.
Okay. So this one is step to like compute medicine.
Yeah, FRBD is having two files. First analysis, second graphical representation.
Oh, okay. So
So this is for C10. Then again, if any other trait we can take. We'll take one. Yeah. So this is for first trait. So likewise here, if we will change the trait name, this type of graphs will come. And now from this data, we can go for PCA. This same FRBD data, we can go for PCA. So this PCA script today I shared, that is same. Output files will, everything will be same, but just the diagrams are somewhat colorful. This will also I will run. Just here I will change the trait name. Yeah. So here you will get the percentage. This percentage were not there in our earlier command. For all the PCs, you will get the percent variance written on the graph. Then this individual PCA graph will come in bit decorative. Then this is characters, traits, variables, PCA. And then this last one, by plot. So this you can go for PCA. Sorry. And simultaneously, you can go for card diagram also. So this card diagram also, you can go with this FRBD data.
Excuse me, sir. For card
Ah, yes.
diagram, do we have a script, R script?
Yeah.
Today?
Yeah. If you don't have, just you can download it from WhatsApp group.
Okay.
It is there.
So from old files?
Yeah.
Okay.
Just click on the name from file. You will get all the commands there.
Okay.
As we have already annotated our FRBD with name data, so no need to annotate it again. So here I don't know the coding for test, so no problem. In 9, red. Then say I'll give light blue. And last 11. So say I'll give light green. It's gray. So directly you can type the name of the colors. So from FRBD data also, you can generate a card diagram. Yeah, Ragini.
Uh, Sir, in grid colors, for C1, C2, we are using different codes, and for C9, C10, we are using color names. Why is it so?
You can use colors for that also. As if suppose now I don't know the codes. So from where you will vest the instead of that directly you can write color name for each and every character. Here also you can change.
Okay, sir.
Hello, sir.
But the bottom line is you can't change the structure of the command. Within that structure you can change anything.
Hello,
Okay,
sir.
sir.
Yeah. Yes.
Please, can you explain the code diagram to those one that are significant or correlated or not?
So see, these legends are visible to you. This
Yes.
1.1 what it indicates?
So one to
What is it?
minus positive,
Ah,
positive and negative correlation.
okay. So for on left hand side, some colors are there.
Yes.
So for what is the purpose of that color?
I think from green, positive correlation, then dark red, negative correlation.
So from dark green to dark red means from positive to
Negative.
yeah. So see, actually earlier or at present also many people are using heat map for correlation, right? But now this
Yes.
representation is there like it is means what you will say it is like one of the form earlier circles was used to be there. So it is like the form of circus. So it is actually circular correlation plot. So it is referred as a chord diagram. So this outer, if you will see the outer layer, you will see the character name that is C1 to C10. There is a names of different variables, whatever we are studying. And whatever the lines are there, these are the lines connecting the two variables. As you can see, some are green lines, some are red lines. One minute, let me plug in the charger.
Yeah, so it's clear now. I was confused with the color that you assigned to different traits, but it's clear now.
Yeah, it is like, it is not like for identification of traits. These are the different colors. That colors will annotate the intensity of correlation among themselves.
Thank you.
So like say this green is indicating positive as you told, red is indicating negative. And whatever the dark and thicker lines are there, wherever you will find the dark and thicker lines, so that dark and thicker lines will indicate the stronger correlation. If it is dark and thick green, so that means significant positive correlation. If it is dark thick red, that means significant negative, and vice versa to the lighter lines. So it is in this way you can interpret this diagram. So in heat map we used to interpret from intensity of color. So here also intensity of color is there, but with intensity of color, these thick and thin lines are there connecting the two variables. So from that thick and thin, this interpretation we need to do. So first we did the FRBD ANOVA. Then after that, we did this mean plot. Then the interaction plots are plots. There it is. This interaction plots was there. Then these mean plots. Then PCA. So this script you can use for your normal RBD also, because these graphs are quite interactive. Just you need to edit the file names and everything. And after PCA, this chord diagram. So this much you can do with FRBD.
Excuse me, sir.
Yes.
Sir, in this chord diagram, region for one variable lies from 0 to 9 in scale.
Your voice.
The region for one variable lies from 0 to 9 as it is marked 0, 3, 6, 9.
You are saying like this outer layer is 0 to 9.
Yes, sir.
Correct?
Yes, sir.
Yeah. So actually, as I told you, this outer layer will represent the different variables. Right? So if you will see, each character will start from 0 to 9.
Okay,
So
sir.
that is actually these are the index. These are the index assigned to each and every trait. Like if we have one trait, so just they divided into 10.
Okay,
These are,
sir,
in other
that means
way you
that if
can
we will
see this
study,
better.
so that means that if we will
So
check correlation between C9 and C10, then for C9 we will study for the region 0 to 9 and C10 also 0 to 9.
Same here you can
We
see
will,
this thick red line is there.
yes, sir.
This line is there if you can see.
Yes, sir. Okay, sir.
sorry, sir. Can you
So
hear me?
if you say this C3, where is C3? C3, C3 to C2, is there any correlation?
No, sir.
C7, so just likewise
Yes,
you
sir.
need to identify it.
Okay, sir.
So likewise, we in D square, we'll say like one genotype from cluster 2 and one genotype from cluster 11. So these two are more diverse. So that means we can use that two genotypes for breeding program.
So, sir, in this chord diagram, the green one is showing like a strong correlation because it's
Yeah.
about to 1, and in this diagram,
So
there's
actually,
more.
color is showing, but whatever the thicker green lines are there, they are showing highly significant positive correlation. So
Positive
as the
correlation.
thickness goes on decreasing, so the correlation value keeps on decreasing with the
Okay,
thickness.
so in most of the diagram, like for the trait, it's showing a positive significant correlation in the
Yeah,
same.
mostly here simultaneously you can find the red thicker lines also. It is less in number, but it is there.
Yes, red are
So
Hello, sir. Hello,
this
sir.
is
Hello. Yes, can you hear me?
yeah. Tell me.
Yeah, still on that card diagram. What is C1 to C11? Those are genotypes, and if I'm not mistaken, C1 to C11 around the yeah.
What is this C1 to C11?
Yes.
Genotypes or what?
Okay, these are you said they are variables, isn't it? Like these are
Yeah,
our traits. These are our traits.
yeah.
Okay, then let's go to the figure. I want to yeah, to that. Yeah. Then the numbers, that scale from 0 to 9 stands for what? 0 to 10 in fact, stands for what?
So these are actually, this is complete one trait, and this 0 to 10 are the components, vector components they have prepared in this diagram. So if you will see for each character, 10 vector, 0 to 9 vector components are there.
Oh, okay. Okay. So let's take for example C10, trait C10. And we see that we have that board green color from around 4 joining to 10. What does that mean? We mean there is a correlation.
Or which joining?
Yes. You see, let's take for example C10, and we are seeing we have a green band. That green color is coming from around let's say 5 there, and it's ended at 10 there. What does that mean?
Yeah, so this thicker green line you are saying, so wherever you will find this thicker line, that means the correlation is strong. That is, in our language, it is highly significant positive correlation.
Within the same trait.
Yeah, within this are vectors.
Okay. Okay. So these vectors are the same vectors that are also indicated using the PC when we using the PC diagram, PCA diagram.
Yes.
Can we?
Here also you can say, na? So this is your PC.
Okay. Okay. Thank you.
This is your genotypes across two PC. Here also you can interpret. So this is everything all about FRBD.
Excuse me, sir.
Yes.
Sir, in this chord diagram, how will we find that these are the significant above p-value correlation is there? This is the only showing correlation values of all things, sir.
Here it is not a traditional model where you will need to give stars.
Okay, sir.
Here this is graphical visualization, and here we need to interpret on the basis of thickness of the color bands. That's it.
Okay.
So here, like, it is not like our traditional where we are seeing the p-value, above p-value 0.5 we are giving one star, 0.1 we are giving. It is not like that. Here just we are interpreting on the basis of thickness of color bands.
Okay, sir. Okay.
So this is FRBD. Now our next is this CRD. So here you will see the genotype, treatment, same experiment data, but for CRD. So here the salinity is not there. And these are the 13 traits. So for betterment, just you can name this, or just you can adjust that space wherever space is there. In some cases, space is there. In some cases, space is not there. So either adjust it or rename it. Anything you can do. Okay, I have adjusted it, so no problem. So this is the TRD script. So first just check it, all the packages. So I will run the library. I will clear this everything. Lots of files are there. Then it is already file name is CRD. So I can import it. Then we need to define the trait. So here also you will get the graphs. So with this script also, you will get these graphs for individual traits. So here no need to run the separate script for this mean plots. So likewise for each and every trait, you will get the graph, and here you will get the ANOVA. So just after that, you will get the why it is not coming. For importing the files in all the analysis in text file, that command you can copy from WhatsApp group.
Sir,
And that, Hmm.
one more confusion was there. So in
Hmm.
generating the code diagram, you once you shared the script on WhatsApp group, sir. In that
Ah,
file name was given traits. For that we have to change our FRBD, or simply we have to paste that.
file name was different. Here we have already annotated, so that's why I have not annotated again.
Okay, sir.
So if we have not annotated, then you need to annotate it. Otherwise, if it is already there, then no need to annotate again.
Okay, sir.
Because if they are everywhere, we need to define the traits column. So file name is traits. Again, we need to define the trait. So the software will not take. वो हिंदी में कहावत है ना, मेरी मुर्गी की एक टांग, ये आर सॉफ्टवेयर वैसा है. So likewise, you will get the text file with all the analysis. Where is the text file?
Excuse me, sir.
Yes.
When we analyze the data, we have some plots and we save it by export. But is there any code? So is there any code so we can use that code and we can just save our
For
the all the plots in one file? Is this possible?
all the plots, let me check in my folder. Let me check. I think it is there. 1 minute.
Excuse me, sir.
Yes. Yeah.
So, sir, the file we are importing for this CRD, uh, in that Excel
Ah,
file, in some of the columns, there is no value, like for genotype 13 and 14. Is it due to randomization or something else?
no. It is the for those blank spaces means observations are not there.
So, like we were not able to record observation for some parameters, or
Yeah,
genotypes were not
yeah. So
performing.
if you can mention it zero also or blank, no problem.
Okay, so like if some genotype is not able to showcase any parameter, then we can let it blank or say, mark it zero.
Yeah, yeah, yeah.
Okay, sir. Okay, sir.
Yeah, please check the WhatsApp group. So with that, directly you can save the output file and all the whatever the graphs at a time in your working directory. So just copy that message and save in text file first. Don't run directly, otherwise you will forget. First save in text file. So just see that this type of folder will come and this total graphs will be there in PNG format. If any system is not supporting that PNG, so just kindly open it with Paint and then save as it with JPG. So this each and every plot is there.
Sir, the one you have sent on WhatsApp, we have to run in the continuation.
So say
Hello, sir.
this whole command is there. Am I audible?
Yes, sir.
Yeah. So this whole command already we have set up the step 2, that is threads to analyze. So directly you can run it. Either you can run it from start, no problem. From start also no problem. Instead of that script, you can use this whole script also, no issue. And apart from this, you can go for PCA and what is that? Chord diagram also. Then other visualization also, that is the density plots and other things we have covered. Sorry. So here, that is same thing, that is QQ plot is there, one graph. And in second graph, that is residual and fitted values. So it is, this representation is somewhat similar to that block.
Excuse me, sir. Your screen
Ah,
is not visible. Like you are saying about
not
something else,
visible.
and
One
then
minute, one
clearly
minute,
reflect.
one minute, one minute. Just 1 minute, give me a minute. Yeah, now it is visible.
Yes.
Yeah. So here see QQ plot, then residual versus fitted. So it is like somewhat similar to the bluff. So this x-axis, these are the fitted value that is predicted by the model. And on y-axis, the actual values, that is residuals are there. That means residual means actual value minus predicted value. So that is called as residuals. And this each point you are observing, this each point is the observation, whatever the our values. So these residuals are actual minus predicted, and fitted means the predicted, and this and on right side you will find the QQ plot. So it will check like whether the distribution of data is normal or not. So if in this case, if your data will follow normal distribution, then all the points should be there in the angle of 25 degree. If your this standard residuals, though these are following the normal distribution, then all the data points should be there in the angle of 45 degree. But here it is not like that. Here several deviations are there. And you can see some outliers outside these lines. Some outliers also it is there. And then in this next, these are the treatment means. So this is one is here you can see this NPBR. So this NPBR is nothing but non-parametric regression. Or you can say that the binary model. So for each and every thread, you will get the graphs and you can see the various types of distribution. So in some cases, data points will distribute normally. In some cases, it will show deviation. One or two data points, maybe it will show the show as outliers. So it is like that. So and then I think we have not seen the CRD. Yeah. So here you can see the analysis of variance for all the tricks. So can anyone tell me what is the use of this LSD test? If you have gone through this text file, LSD test is there. So what is this LSD test?
Least
Least
significant
significant difference test.
What is the purpose?
And actually, two will be there, LSD and DMRT, like that.
Yeah.
In
So,
DMRT, they are using in entomology that
yeah.
that insect counts and all. They will go for that. For our field parameters, we are using LSD.
Yeah. No, but what is the exact purpose of this LSD? Why we are using here?
Is it for separating means?
Mean comparison, sir.
Yeah. Yeah. So mainly this LSD we are using for comparison of means. So from that comparison, we can conclude like which specific treatments are significantly differing from other. So if you will see for all the traits, LSD test results are there.
Or we use this one least significant difference for at par varieties when we do analysis or not?
So everything it is, see, wherever you will find, see this analysis, everything they are giving. So in CRD, we are running this LSD for just comparison of the means for significant treatments. So this is the output file, and then so just for other the aspects also, by using this command, whatever we did here, so we can design something like this to export all the figures at a time, or maybe it is available. I need to see. So like here we have exported all these 13, 14 graphs at a time. Yeah. And other thing, what we did this conversion today, HapMap file conversion into numerical, that we got two files, SNP information and numerical HapMap. And along with this phenotypic data, so all these three files we will deal tomorrow for QTL analysis, for GWAS, and for calculation of blobs and blue. So we'll calculate trait-wise blobs from raw data, not from this whatever I am sharing, because this is already calculated. So I will share raw data, and from that raw data, how to calculate blob and blue. So that thing we will see. Now I think only alpha lattice is pending from our side. Majorly we have covered RBD, augmented FRBD and CRD.
And so diallele,
Yeah,
split
diallele
plot.
split plot is there. Diallele we will cover after this, madam. We have separate section for heterosis combining ability.
Okay, sir. And
In that we
you
will
say like
see
genomic
full
selection
diallele,
will
full
do.
diallele, half diallele, line into tester, everything. When we
And
need
what?
to go for full diallele, when we need to go for half diallele, which one is better?
Okay. And you said like about genomic selection, we have planned to do.
Genomics, that is only by calculating blobs and blue. After that, we will go with that same blobs and blue, then we will predict the
Okay.
genetic potential.
Okay.
Haji, Doctor sir.
Yes, sir. Excuse me. Another thing, please include as already discussed for single replication data, either augmented or multi-location trial, how blue and blob can be used.
For
They,
augmented, na?
for augmented, sir.
For augmented, already we have raw data. From that raw data, we can calculate the blobs tomorrow, no problem.
Okay, sir. Thank you.
Yeah,
And
I
sir,
have already shared.
Yes, sir.
The augmented raw data with you.
Yes, sir. And another
Yeah,
thing
so,
is that it needs a video. The two session we have actually missed. So
just wait for 1 minute. I will call Vivakar sir. 1 minute.
it is very difficult for us to practice. Actually, if the video is uploaded soon, I think.
1 minute, 1 minute. I will call him. 1 minute. Give me a minute.
Sir, one more thing, sir.
एक मिनट रुको राज, मैं कॉल करता हूँ, बाद 1 मिनट में आता। हाँ राज,
Sir, that CRD that we did today, sir, Just can we do that revision tomorrow, sir, once again for some time?
Yeah.
Sir, from starting, sir, from running the script, sir.
No problem.
Okay, sir. Thank you, sir.
Yeah, WhatsApp group. Actually, we have already one group of plant breeding insights. WhatsApp group is there. There we can. It is open to all.
Sir, can you please share the link for joining that group?
Yeah, 1
Sir, which
minute.
group?
It is open to all. You can discuss. You can discuss there. No problem.
Excuse me, sir.
Pawanji.
Sir, I am not getting that CRD ANOVA in working directory.
So check the WhatsApp group. उसमें मैंने कमांड लाइन भेजा है रिसेंट वाला.
Okay, sir.
उसको रन करो. पूरा का पूरा ही फिर से रन करो. कोई टेंशन नहीं.
Please, sir, if you can upload all the videos, I think it's going to be better because the last video we have on the site, I think is the day 10 video.
That person is not reachable at the moment. I don't know. Yeah, he is now, I think, out of network. He will reply to me, no problem. So that WhatsApp group link I have shared in this 45 days group. So those who are not there, just join. That group is open. There you can discuss. There you can discuss the things. Many members are already there. From this batch also, I think many members are there. So those who are not there, just join that. Here you can.
Excuse me, sir.
Yes.
Sir, you told us to download structure software, no, sir?
Yeah. That also we need to. This molecular, it is very big. We need to cover it.
Which one should we download, sir? There is graphical front end and without front end, and analysis for Gina
With graphical. With graphical.
with graphical front end.
Yeah.
Okay, sir. Thank you, sir.
And apart from this, keep in your system QTL cartographer also. I think version 2.3 or 2.5 something. QTL cartographer.
Okay, sir. That one I downloaded.
Yeah, everyone just download it.
Sir, could you please maybe write it down in the WhatsApp, the type of software that we should download, cartographer and the version, please?
I will share the link. I will share the links directly.
Okay.
So that it will be easy for everyone.
For sure.
Sir, when will we learn the split plot design?
Split plot. Okay, that is, we can cover in between these sessions, no problem.
Okay, sir.
Split plot and split plot, alpha lattice. So only two are there. Rest we have covered it. No issue. Because for some members I have shared lattice script also. I don't know whether they have done that successfully or not.
Excuse me, sir.
Ajit.
Sir, can we run chord diagram and PCA for RBD data?
Yeah. PCA, so we used to run. You can draw a chord diagram for RBD also. Instead of that heat map, you can go for chord diagram.
Okay, sir.
You can go for chord diagram, then density plots also. You can go for this FRBD, CRD. Wherever you have replicated data or mean data, you can go for this visualization methods. No, it is not number related to number of genotypes. Actually, it is giving us the interrelationship between the characters from that data.
Okay, sir.
If you have alpha lattice data also, for that also you can go for chord diagram. Because in alpha lattice we used to screen 500, 400, 700 more number of lines.
Okay, sir.
So in that plant breeding insights, you can, it is open to all. You can post, you can ask, you can discuss. Anyone can post if you will find anything new on any social media platforms or from any different sources. Just you can, if you want, you can share with everyone. So this, please ready with this software. Tomorrow we will start with this everything. Okay. In meantime, I will continue to try this Bibakar sir's phone. I will tell him regarding the videos. Okay.
Sir, for chord diagram also, if there will be, sir, that script that you shared on in library, sir, same we have to copy and paste for any of the file, or we
Yeah,
have to
but
change
just
it?
you need to see the file name, you need to see the number of traits, names of traits. So there you need to edit it. And you need to see whether after editing the traits, just you need to see the rest of the commands. It is
Okay,
not
sir.
like for each and every file you can blindly use that command lines.
So, like here it is written PHDTF. In that we have to write C1, C2, and till C11. And for color change, you said that we can type the color name also.
Yeah, you can directly type the color name.
Okay, sir.
Instead of coding, yeah, Dr. CL, Sarji, are you there? CL sir. I think he is not there.
Sir, cartographer software link, if we could get, that would be very helpful.
Cartographer, 1 minute, wait. I will send to you. By afternoon it was not working, but just you can check it. So that links I will share in WhatsApp group so that you just download it for each all these 3-4 softwares what I told. The link I will share. Okay.
Okay, sir.
Yeah.
Excuse me, sir.
Yeah. I think
Sir,
yeah,
for
you have.
sir, for LSD also we have to reach the data in the same
So
manner as we
actually,
did for CRD.
arrangement is almost same, but some slight changes are there. I will share to you, no problem.
Okay, sir.
Yeah, I will share on your WhatsApp number.
Okay, sir.
Okay. So thank you guys. Tomorrow 6:15 we will complete our pending this association mapping part. Okay. And regarding that videos, I will update you before I will before 10 or 9:30, 10 o'clock. I will update you.
Okay, sir. Thank you.
Okay. Thank you. Thank you, everyone.
Thank you, sir.